Today we're going to be talking about the process of transcription. And transcription is one of the two processes that we're going to need to undergo from once we have our DNA, right? And we want to make proteins, right? Protein synthesis is what all cells are trying to carry out. So we can't just take that DNA and make a protein with it. We have to go through two steps first. The first of that is the process that we're going to be talking about today, which is transcription, where we're going to turn that DNA into RNA, right, an mRNA transcript. And then once we have that mRNA transcript, then we can undergo translation, which is where we actually get our protein. But today, we're going to focus on transcription and how we go from having a DNA molecule in the cell with all of its information and then creating an RNA transcript, which is able to then go out and execute these orders and make a protein. So transcription converts DNA into RNA. And anytime we're dealing with DNA, we're going to be working inside the nucleus because the DNA of a cell is never going to leave the nucleus. And there's a couple different reasons for that. DNA is an extremely large molecule. It's very large, it's very cumbersome, it is tightly wound, tightly bound, and it's just kind of crammed inside the nucleus of your cells. In most cells that have a nucleus and have DNA inside your body, there's, even though those cells are microscopic, you have over 3 feet of DNA inside of that cell. So there's an enormous amount of DNA packed in there. So we don't want to take it out of the cell's nucleus because it's just too big. The second reason is that DNA is really important, right? DNA is the instructions for life. So we don't want to risk taking DNA outside of a nucleus when it doesn't need to be, right? So we're going to keep it safely tucked away inside the nucleus. An analogy I use is to think of it like this. You have the building plans, the blueprints for a large building. Let's say it's a 50-story building, okay? And you have all the blueprints for that entire building, the electrical, the plumbing, the concrete, where the doors go, all of those different types of things. You are going to keep those blueprints tucked away somewhere safe, back at the architect's office, right? And it's a very large document and it's there. If someone who's working on the 37th floor comes and says, I need the electrical wiring plans for the 37th floor, they are not going to hand him the entire blueprint for the entire building, right? Because he doesn't need them. He doesn't need to know what's going on the 17th floor, or the first floor, or the 50th floor. All he needs is the 37th floor. So the architect would hand him the blueprints for the wiring on the 37th floor. This is really a lot like what happens in the nucleus, okay, with DNA. DNA is full of genes, but we only need one gene. So when we make an mRNA transcript, we're only making a transcript of one particular gene, right? So that way the DNA can stay housed in the nucleus, just like the blueprints for a building can stay housed back at the office. If a person takes the blueprints and takes them to the job site and they get torn up or they get coffee spilt on them or whatever, they can just go back to the office and get another copy. That main copy stayed safe, unaltered. That's what we're doing with our DNA and RNA. The DNA stays safely tucked away. If something happens to the RNA molecule as it leaves the nucleus, something in the cell, it doesn't make it to where it's supposed to go, that's okay. We go back to the DNA, make another transcript, no harm, no foul. So the purpose of transcription is to give us a copy that can leave the nucleus because the DNA is not going to leave the nucleus, right? The RNA transcript, very small, slips right out of the nucleus into the cytoplasm of a cell. And then from there, it's going to go find a ribosome and undergo the process of translation, which is the final step. Again, DNA, very, very large, very, very important, not going to leave the nucleus. So anytime we're talking about something involving DNA, it's got to be taking place in the nucleus, which is exactly where transcription takes place. This process is going to resemble DNA replication. Remember, with DNA replication, we're matching up base pairs, complementary base pairs from one side of the strand to the other. So if there is a T over here, then we're going to provide an A there because that's a complementary base pair. If there's a C here in DNA replication, we would provide a G nucleotide to be complementary. Okay? The only difference is when we're making RNA, we're not going to see that T, which is a thymine. Instead, if we have an adenine base pair, when we're building a complementary transcript of RNA, we're going to replace that thymine with U, which stands for uracil. So if you're ever looking at a strand, a molecule, and you don't know what it is, if it has a U in it, you know that it's RNA. If it has instead a T in it, you know that it must be DNA, because you're never going to find that uracil in DNA, and you're never going to find that T in RNA. So again, it's going to look like DNA replication in the fact that we're adding base pairs and they are complementary. So it happens at many points on the strand at once. So along the DNA, we're going to be doing this process. And only a short part is transcribed. That part that we are transcribing is the gene itself. But there are other parts that are needed for other processes that we're going to kind of see here as we go through this. So base pairs, base pair ruling still applies. We're going to substitute uracil for thymine. The first two DNA strands, obviously, before we get started on this, we're going to have to separate the strands. Okay, this is where the process begins. Only one side of the strand is going to be used. So one side of the strand is going to be used as the template. The other side isn't going to do anything. So once we separate things, then we're going to kick things off with a molecule called RNA polymerase. RNA polymerase is an enzyme. We know that it is an enzyme because on the end of that word is ASE, and ASE is a suffix that means enzyme, just like the suffix OSE means sugar. So we have RNA polymerase, which is an enzyme. It's going to attach itself to the DNA molecule, and it's going to add on the nucleotides. Okay, the binding to the strand is part one of the transcription, and this is called initiation. It's where we start. Okay. So this RNA polymerase is going to allow the nucleotides to simply be added to RNA. A promoter sequence, which is a specific order of nucleotides, tells the RNA polymerase where to bind and where to begin this transcription. Okay. The second phase of transcription is called elongation. This is where the RNA polymerase is going to be adding the base pairs. Okay. As more of the RNA strand is synthesized, it peels away from the DNA template, which allows the two DNA strands to come back together. So we're going to add those base pairs, right? DNA is separated. We're going to add the base pairs. As it moves this way, DNA is going to come right back together, kind of closing in behind it. As each new nucleotide is added, they're going to form hydrogen bonds, which hydrogen bonds are the same bonds that hold two strands of DNA together. Okay. And those bases, the bonds are going to be held there, and this forms them into a strand of RNA. The third phase of transcription is called termination. The RNA polymerase reaches a terminator sequence, which is basically telling it to stop. It signals the end of the gene, and it's going to detach from the RNA and detach from the gene. You now have an mRNA transcript that is ready to leave the nucleus and go find a ribosome. The next stage or the next step is where we're actually going to get the production of a protein, and that is, of course, called translation. But all this has to happen before we can ever get to that process of translation. We cannot take the DNA out of the cell. We can't, just extract a gene and leave it in the form of DNA. Messenger RNA is our vehicle for allowing this process to happen. Without messenger RNA, we don't have the means of getting the decoded message that was on the DNA outside of the nucleus. And again, this process of translation has to occur at the ribosomes, and so we've got to have a way of getting it there. We can't just have everything happening in the nucleus, right? Again, we want to protect that DNA. We want to kind of keep it there and allow, because it's going to be sending genes throughout the cell to all different locations. It's going to be, ensuring that every single cell has the proteins that it needs. So, you're going to have RNA transcripts shuttling out of the DNA constantly, out of the nucleus constantly, to ensure that all of the genes are being produced. Because this just isn't just happening, once a day or anything like that. We're having thousands of genes that are needed to be transcribed to produce all the proteins that we need and that all organisms need to maintain life. So transcription, again, is just the first process where we're going to take DNA and we're going to transcribe it into an mRNA transcript. I'll see you next time.